This PowerPoint will help you understand what you need to know about inferential statistics in order to be able to appraise studies in the scientific literature. Now, as I mentioned in the previous PowerPoint, there are three basic categories of statistics that we're going to talk about in this class. The first is descriptive statistics, and we've already seen a PowerPoint or a slides presentation on descriptive statistics. Remember, with descriptive statistics, you're basically describing the data. You can describe this data and condense it using a chart or a graph. And remember, it's usually in some form of proportion or percent. With inferential statistics, you are going to be able to make predictions or inferences from the data. With the inferential statistics, you take your data from samples and make generalizations about a specific patient population. One of the main features of inferential statistics is going to be hypothesis testing. This is where you can use sample data to answer your research questions. The rest of this PowerPoint is going to be spent describing how to interpret inferential statistics. So as I said in the last slide, inferential statistics are often used to answer a research question. What I'd like to do is go ahead and give you a research question and kind of walk you through how inferential statistics can help answer this question. The research question is shown in this slide, and that is, in patients with diabetes, is aerobic exercise more effective than lifting weights for controlling blood glucose levels? So once a researcher has formulated a question, they start with something known as a null hypothesis. The null hypothesis is simply there is no difference between the two treatments. And the two treatments in this research question, if you remember, are aerobic exercise or weightlifting. So the researcher's null hypothesis, stated another way, as written here, is the difference between aerobic exercise and weightlifting for blood glucose levels of patients with diabetes is zero. There's absolutely no difference between doing any of those things. That's where you start. Next thing to do would be to perform the experiment. And when you perform the experiment, what you do is you take a group of people and all with diabetes and you split them into two different groups. You can do it randomly, so make it a randomized control trial. And you have one group of people perform aerobic exercise, you have the other group of people do some weightlifting, and over a period of time, say 6 months or so, you check to see if there's a difference in their blood glucose level control. Well, the question is, after you do that experiment, how do you know if there's a difference? In other words, how would you know that the null hypothesis was really false? And the answer to that question is you do inferential statistics. The job of inferential statistics is really to rule out the possibility that the result that you got was due to chance. To further explain that, I'd like to just start out with something really simple, and we're going to look at the role of chance. If you took a coin and you flipped it 10 times, you would expect about 5 heads or 5 tails, right? So the chance of getting a heads or a tails is 50-50. If you flip the coin 10 times, you would expect 5 heads and 5 tails. But you flip the coin a number of different times in your life, and you don't always get 5 heads and 5 tails when you flip a coin. So how would you know that the coin wasn't rigged? Let's say that the coin was weighted heavier so that the head side or the face where heads would be displayed was a little bit heavier. So every time you flipped it, you'd get more tails than heads, or vice versa. How many times or how many heads and how many tails would you have to get when you flip the coin to know that the coin was really intact, or the integrity of the coin was intact? So let's talk about inferential statistics in the simple experiment of flipping the coin 10 times. So let's do that experiment. We're going to do an experiment to test the integrity of this coin. The null hypothesis, going back to inferential statistics, would say that the coin is not rigged. That means there's no difference between getting the number of heads and the number of tails that you would get if you flip the coin 10 times. So you flip the coin 10 times, and lo and behold, you get all heads. So that sort of makes you think something weird's going on. right? I mean, usually you get at least one tail. We don't know how many tails you get, but you think maybe one tail in 10 times. Well, what's the chance or the probability of getting all heads if you flip a coin with two sides 10 times? And it turns out that the chance is less than 1 in 1,000. Another way to say that is 0.001. So as far as inferential statistics is concerned, well, this is called a p-value. If you look, p here, the italicized p would be, or the p, or the probability of getting all heads from a decent coin is less than 0.001. Okay, so should you conclude that the null hypothesis is false or that the coin is rigged? And what kind of p-value would you need to say that the null hypothesis was false? And it turns out that the standard conventional p-value to say that the null hypothesis is false, or there really is a difference between two things, is 0.05. So if p is less than 0.05, this is considered statistically significant. Another way to say, or just another term to say that something is statistically significant, which is less than p 0.05, is this is also called alpha. And alpha, by definition, is nothing more than where the researcher has set the level for statistical significance. And as I said, by convention, it's standardly p less than 0.05. Now, go back and look at the results from the experiment that we just did. We flipped that coin 10 times and we got all heads. So since the probability of that happening is p less than 0.001, 0.001 is much less than 0.05. So we can accurately say, or say with pretty good confidence, that the null hypothesis is false and the coin is actually rigged. So in order to prove that there's something wrong with that first coin, we need to test the experiment again with another coin. So we're going to go ahead and test the integrity of another coin. And again, we're starting with the null hypothesis that the coin is not rigged. So we go ahead and flip the coin 10 times. First time we flip it, we get 5 heads and 5 tails. Okay, that p is equal to 0.5. And that would be expected. So what you'd expect is you'd get that 50 times out of 100 coin tosses. So since p is equal to 0.5, that's looking good, right? You think that we can accept the null hypothesis. There's really no difference. There's no problem with the coin. So we thought, okay, let's do it again. Same coin. Second time we get 8 heads and 2 tails. That would be a p-value of 0.11. Again, 0.11 is greater than 0.05. It would be expected 11 times out of 100 coin tosses. And since that number is greater than 0.05, we're still good. We're still going to accept the null hypothesis. We do it a third time, and we get 7 heads and 3 tails. This is a p-value of 0.34. Again, from this experiment, this is obviously a much better coin, and we would accept the null hypothesis. And as far as interpreting p-values, this is all you need to know. You don't need to know how to do the statistics to get the p-value. You just need to be able to look at a figure or a table that's coming out of a manuscript and interpret it, saying they should accept the null hypothesis, meaning there's no difference between two treatments, or they should reject the null hypothesis, meaning that there's a big difference from one treatment to another. And again, the significance level is set at alpha at p less than 0.05. Okay, now that you've learned a little bit about p-values and how to interpret them, let's go back to our original research question. If you remember at the start of the PowerPoint, we were looking at two different groups of individuals. Our research question was, is there a difference between aerobic exercise and weightlifting on blood glucose control in patients with diabetes? So what we did is we took patients with diabetes. In this case, the number is 804. We separated into two groups. 403 were in the aerobic exercise group and 401 were in the weightlifting group. We waited about 6 months, and then after 6 months of time, we measured the blood glucose, and we decided that if the blood glucose was uh, greater than 100, that wasn't such a good thing. Okay, they didn't have very good blood glucose control. It turned out that in the aerobic exercise group, 123 people had blood glucose that was greater than 100, which is approximately 33%, and 153 people had blood glucose that was greater than 100 in the weightlifting group, approximately 37%. Now, in the absence of having a p-value to help you interpret that, you think it's pretty close. You might think that there's a difference. Maybe, I mean, what is 4 percentage points, right? Is it significant or not? Well, it turns out that the p-value is 0.11. And hopefully you're thinking, well, since the p-value is greater than 0.05, I'm thinking that there is no difference and that I should accept the null hypothesis. If you said that, good job, you're correct. Believe it or not, there's actually a time when you don't want your p-value to be less than 0.05. And that is at the start of a research study. Let me see if I can clarify that for you. So I have two groups of patients. We can just go back to our diabetic patients, and I had one group that was in the aerobic exercise group, and then one group that was in the weightlifting group. Let's say before the start of the study, I want to look to see what the average glucose level was in the aerobic exercise group and compare that to the average glucose level in the weightlifting group. Before I do any kind of intervention with this patient population, I want there to be no difference between the two groups. So at the start of a study, when you're looking at the baseline demographics of the individuals who started the study, you want the p-value to be greater than 0.05. Remember, being greater than 0.05 tells you that there's really no difference at the start of the study between these two patient populations. And just as you saw with the descriptive statistics, it's the same thing for inferential statistics. And that is the type of the data dictates the type of statistics that the researchers will use. You do not have to know what kind of statistics to use. You just have to be able to say, yeah, it was a good choice by the researcher. And if it's nominal data, they're using something called chi-squared. Don't know how to do chi-squared, don't worry about learning how to do any of these, just recognize the name that goes with nominal. Ordinal, Mann-Whitney U, etc. I mean, all of these are some just different kinds of statistical tests. Again, notice that there's a difference between normally distributed data and not normally distributed data as far as continuous data or numerical data is concerned. Okay, just this is for your information so that when you see these words or this set of vocabulary, you know what it means. Almost to the end of the inferential statistics. One more thing I need to introduce you to is that a researcher can correct for multiple statistical comparisons. The only thing that they have to do is something called a Bonferroni adjustment. And what that does is it helps to avoid a type 1 error by adjusting the alpha level or the significance level. And all you have to do is divide the alpha by the number of tests that they're performing. So if they have one data set and they're performing a number of different statistical sets on this data set for comparison, they need to have performed a Bonferroni adjustment to account for it. That's it. Congratulations. We made it through inferential statistics. Lots of information in this slides presentation. What I'd like to end with is a cartoon, and I thought it was really funny. And it's a nonlinear pattern with outliers, but for some reason, the researchers are really happy with the data. And you can see the nonlinear pattern as the smile and the outliers as the two eyes. I guess it's just something that nerds appreciate, so I thought it was funny.